Welcome to Can We All Agree? In our first episode, we looked at good reasons we have to consume media with a healthy dose of skepticism. Of course, when a pandemic that mystifies experts drastically changes life for us all, and people are being barraged with mixed messaging, we end up with masses wondering who they can trust. Just kidding. Who am I to be talking about COVID origins? You should be skeptical of me as well. Well, I'm a teacher, and I've taught and written biology curriculum for up to college level students, but I'm not an expert. I'll show the research as I discuss it, so I welcome any actual experts to let me know if I misunderstood anything in the comments below, because it was very complex, and I promise to follow up with a video making any corrections if necessary. I want to focus specifically on what problems we can identify that could have led to this pandemic, and what we could do to prevent similar problems happening everywhere from causing even worse future pandemics. We have good reason to have a lot of questions about how this pandemic emerged. First, there's the nature of the beast itself, and then we have human nature, racism, nationalism, and the big money of biotech, all clouding our understanding. The first aspect that makes this virus so difficult to track is that 40% of those infected feel no symptoms at all. 60% of the spread comes from asymptomatic carriers. So if you're feeling awful and you're not a terrible person, you'll probably take precautions to guard others from getting what you've got. However, if you don't feel sick, you're not going to be taking those precautions. Then, if you take a test and that test comes back negative, you'll take few to no precautions. Unfortunately, there's a range of false negatives of anywhere from 2 to 29%, depending on the country. The test the White House was using was inaccurate up to 47% of the time. Finally, there's the fact that COVID is an RNA replicating virus, and RNA viruses mutate at a rate 1 million times faster than our human DNA does. There's a really cool open source project shown at nextstrain.org that includes collaboration among over 200 research centers around the world, where they are tracking the different strains of COVID they find so we could see how it's evolving over time and place. Just looking at this part from December 2019 to March 13 of this year, tracking 169 strains. Now, many of the mutations don't really amount to a change that's significant. However, the color changes we see here show times when there was a structural or behavioral change in the virus. So we see about 12 significant mutations in the virus just over a period of 3 months. So we can't really know how much it could have been mutating among asymptomatic carriers before it finally made someone sick enough to actually have to go to a doctor. Then that doctor was able to identify it as something new, not another flu or pneumonia or bronchitis. Then they were able to test for and accurately identify what it was. So all of these factors make it hard to pinpoint a true patient zero. And they're also why it's really important for us to wear masks so we don't give the virus more chances to keep mutating into something that could kill even more victims. Just as we depend on people to minimize the spread of the virus, our knowledge of the virus is based on the honesty and awareness of the possible carriers of the disease, scientists who could be responsible, and government officials who could be responsible. These people are not perfect government or science robots. They're human beings prone to all the frailties that come with being human, including human error. When they make mistakes, they might lie to cover them up or not even be aware of them. No matter how careful we are, we all run on autopilot for part of the day, especially with the most repetitive behaviors we do. That's why we're more likely to get into car accidents when we're close to home. Unfortunately, in order to deflect responsibility for the pandemic in the United States, government officials pointed blame to China and Chinese people, which led to 2,120 reports of hate crimes against Asian Americans just from April to July of this year. 832 in California alone, 81 of which included assault. And that's just what was reported. Then the Wuhan virologist who's been working with bats and coronaviruses for over a decade was receiving death threats. So in addition to individual people being targeted, we also have geopolitical complexities balancing upon a history of war and conflict among different countries. We try to manage these with agreements on what is and isn't okay in times of peace and war due to the shared global threats. These depend mostly on an honor code system and self-policing, regulating, and reporting. It's another reason why placing blame is dangerous, because we need all participants to share as much honest information as possible. Now, these agreements advance with our technologies and warfare capabilities to include guidelines on epidemiological studies, as a lot of what can be studied to prevent a pandemic can also be used to start biological wars. So when we try to designate what level of security is necessary for different laboratories working with different pathogens, the biosafety level 4 is the highest level of risk, dealing with life-threatening airborne pathogens that have no treatment and present a global risk. I heard a conspiracy theory about the lab origin of this pandemic pointed to an academic journal. And that's odd for there to be any credible support for a conspiracy theory, so I looked it up. And I found SARS-like cluster of circulating bat coronavirus pose threat for human emergence. You can see this title in Nature Medicine. I'll put links to all the research I found in the About section below. So there were 17 authors involved, including researchers mainly from the University of North Carolina, Chapel Hill. There were 12 total authors from there. One from Harvard, one from the FDA, one from the Institute of Research in Biomedicine in Switzerland, and 2 from the Wuhan Institute of Virology. So they discuss recent metagenomic studies they've been doing where they go into these bat caves and collect saliva, blood, and feces from different bats to see what kind of microbes are in there. And not only what kind of bacteria, viruses, and the like they can find, but even the genetic particles of some of those microbes. So then they take all this data and they map it out so they can see how different pathogens are evolving over time and prepare for if there's some kind of viral gene mutation that will make anything that is infecting the bats transmissible to humans. This data can get really noisy, but fortunately now we have artificial intelligence and logarithms to make sense of it. However, as they say at the end of this quote, sequence data alone provides minimal insights to identify and prepare for future pre-pandemic viruses. So they say that metagenomics isn't enough. Their solution? To examine emergence potential of circulating coronaviruses, we built a chimeric virus that encodes a novel zoonotic spike protein in the context of a viable coronavirus backbone. Some of these words might look familiar to you from the current pandemic. A chimeric virus is kind of like a mule where there's joined nucleic acid fragments of 2 or more microorganisms. So that can happen if one host is infected with 2 or more diseases or in a lab. Zoonotic means that it's contagious among different species of animals, like rabies. So they infect a mouse and pass that infection to another mouse, etc. 15 times, so it has time to naturally mutate within its host. And then it gained the functions of elevated replication in our lungs and lung pathogenesis in aged mice, which is again similar to the current virus. They continue to explain that due to 6 coding genetic mutations associated with the mouse adaptation, it will more effectively be able to replicate in human cells. They also tested it in living lab-grown human lung cells. And they explained that creating this mutation plus the metagenomics data helps them predict and prepare for future emergent viruses. I looked up the researchers and a lot of their past studies. All of them have been researching similar epidemiological studies. So reading this academic journal and looking up the history of the researchers involved convinced me that there could possibly be a connection to this study. However, I don't believe that there was an intentional leak. I also heard experts were saying that it wasn't possible, so I looked up the research rebutting it. I found no credible evidence supporting claims of the laboratory engineering of SARS coronavirus. Here they address rumors of a direct leak from their lab where a strain of bat coronavirus has 96% homology with the one causing COVID. That means 96% of the genetic sequencing of the coronavirus they have in their lab matches the sequencing of the one causing the pandemic. They continue to say, given that there are over 1,100 nucleotide differences between the two strains, distributed in the normal pattern of the virus's natural evolution, that it's highly unlikely that their coronavirus is the immediate source of the one causing the pandemic. I emphasized the words directly and immediate because those stood out to me as the scientists doing the research trying to responsibly show that there was the possibility of an indirect connection. Then they reference a claim of connection through the 2015 study we review. They respond, however, this claim lacks any scientific basis and must be discounted because of significant divergence in the genetic sequence of this construct, with a new SARS coronavirus 2 of over 5,000 nucleotides. So, the one that they have in their lab, that's 96% similar, still has 1,100 nucleotides of difference. And the one from back in 2015 has over 5,000. However, if we look at this study from August 19, 2020, we see that in under 4 months, there have been 1,516 nucleotide level variations just in the COVID-19 viruses found among humans. So I think it is possible for something to have gotten out of the lab and mutated enough nucleotide variations to have turned into what we're facing in the pandemic today. What does our famous Wuhan bat lady think? In an interview in Scientific American from March 11, 2020, we see, if coronaviruses were the culprit, she remembers thinking, could they have come from our lab? She frantically went through her own laboratory's records from the past few years to check for any mishandling of experimental materials, especially during disposal. She breathed a sign of relief when the results came back. None of the sequences matched those of the viruses her team had sampled from bat caves. That really took a load off my mind, she says. I had not slept a wink for days. Now, this was updated in June, and you can only find the original interview in archived pages. Now it talks about how there were no infections found among lab personnel, but it doesn't say anything about any antibodies that could have been found. I don't know if the original interview is no longer available due to Chinese pressures, to avoid hate crimes, or to avoid more behavior that interferes with finding the best solutions. However, I think it's important to see that even she was acknowledging that there was a possibility that it could have gotten out of their labs. I think it's also interesting that she specified disposal, as did this article in Business Insider of April 14, 2020. The article states, to be clear, there's no evidence to suggest that the novel coronavirus was created in a lab as a bioweapon, but the newly reported diplomatic cables shine a light on a theory that the virus may have leaked out of the lab by accident or through bio-waste, infecting the surrounding population. So they specify a lack of transparency of government review and approval processes, serious shortage of appropriately trained technicians and investigators needed to operate this high containment laboratory. When they were mutating coronaviruses to be able to infect mice, they were still operating on a biosafety 2 or 3 level. But at the time of this cable, they had just built a wing that was the first biosafety level 4 lab in China. Finally, the staff there welcome more help from U.S. and international organizations. There was already some staff from the University of Texas helping to get them to achieve the highest standard. They said they wanted to achieve an international gold standard of biolab safety. The cable goes on to warn about the coronaviruses that have been adapted to infect humans that they're studying there. So they were worried about that getting out. I looked to see if I could find the actual cable, and I did on the government's website. And we can see here that it was sent on January 19, 2018 from the American Embassy in Beijing to the Secretary of State in Washington, D.C. So usually we have one Secretary of State per presidential term. However, in 2018, we had 3 different Secretaries of State. Trump said he prefers acting directors, but these are the life-threatening matters that can slip through the cracks with this much turnover in important positions. So we knew back in 2018 that a leak was possible from this lab and they had coronaviruses that could infect humans. We don't know if it ever did leak out of the Wuhan lab, but we do know it did leak out of the university in North Carolina that collaborated with them on making it. There were 6 accidents that we know of. A mouse that was infected with a coronavirus that was adapted to be able to infect humans bit a researcher on the finger in 2016. She wasn't told to quarantine. She was just told to wear a mask for 10 days while in public and at work. So we also knew that masks worked in preventing the spread back then. In addition to their coronavirus accidents, the University of North Carolina had 28 incidents reported, according to the National Institute of Health, from January 2015 to June 2020. Now they've spent $480,000 in 2018 and $595,000 in 2019 in lobbying, which may interfere with any government oversight of their operation. The National Institute of Health told the university that they had to cooperate with the journalist in revealing details about the virus involved in the accidents. However, they refused, and National Institute of Health, CDC, and UNC officials all declined to discuss the risk to the public in 2016. They're not the only ones making mistakes. An Army lab in Utah accidentally shipped live anthrax to 194 labs. The Pentagon reported that it was sent to facilities in 50 states and 9 countries. This happened in May 2015. It was supposed to be radiated first, but they were radiating too much at a time, not testing it adequately, and at least 52 recipients identified living anthrax cells that they got in the mail. This is all pretty paradoxical since a lot of biolabs got the green light on this kind of testing because of the anthrax scares back in 2002. Allison Young is the reporter who was pushing for the information, and she's an excellent journalist who's been exposing many biolab risks that have been hidden from the press and Congress. I tried to look up the whole project, but that link was broken. You can find it on webarchive.org, and you can find each of the individual articles. Her reporting has led Congress to realizing that they weren't getting all the information they were supposed to be getting. One article talks about how they delayed incident reports, citing that 2002 bioterrorism law. Then they heavily redacted reports. Using the Freedom of Information Act, she pushed the CDC to release reports of biolab accidents that had omitted info to Congress twice in 7 months. She also pushed universities to honor transparency conditions. She talks about 18 different facilities that tried to charge money for the copies, redact them, reference unavailable documents, ignore her requests until a formal National Institute of Health complaint was filed, and give conflicting reports in oversight. There have been accidents with Ebola, a deadly strain of bird flu, reconstituted 1918 pandemic, and the bubonic plague. The New York Times discusses how documented federal laboratory mistakes with anthrax, the flu, and smallpox are just a tiny fraction of the hundreds in recent years amid a sprawling web of academic, commercial, and government labs that operate without clear national standards or oversight. From 2004 to 2014, we went from having 400 of these high-level containment labs to about 1,500. The Government Accountability Office alerted Congress that the more of these we have, the greater the chances of dangerous blunders or sabotage, especially where oversight is so fragmented and largely self-policing. The Bulletin of Atomic Scientists tries to track man-made threats to humanity. They specify how human error in high biocontainment labs is a likely pandemic threat. In this article, they talk about how they can calculate the risk of human error possibly leading to a pandemic based on the different incident reports they received from the Federal Select Agent Program. They looked at the reports to see which incidents were caused more from human error or equipment failings and the like. Here we can see some of the factors that we discussed: a fear of informing the boss after exposure, not respecting certain biosafety practices, lack of attention, and not enough training. They determined that 16% chance of human error causing a pandemic because they were looking at 14 laboratories that contained a bird flu that could infect mammals and that had airborne transmission among those mammals and could kill up to 60% of the population. that it infected. In comparison, the 1918 pandemic only killed about 2.5% of those it infected. Now, this kind of research is called gain-of-function research, where they look at a pathogen that's already deadly and see how they can make it deadlier or more contagious, just because that could happen in nature. And if they can do it in a lab, they could be better prepared to deal with it if it does happen in nature, is what they think. Now, considering all the biolab accidents that had been in the news and the thought of the 16% probability of this one getting out over the next 5 years, over 200 scientists came together and were like, whoa, we need to stop this. Now, in 2011, there was this realization that, we're mutating viruses to become more deadly, and then we have to publicly publish this research because it was publicly funded. Do we really want to give the instructions to the public on how to mutate a virus to be more deadly? The decision was that if it was publicly funded, it had to be available to the public. Now they could privately fund some research if they don't want to expose it to the public. So there was a moratorium on US funding on this kind of research from 2014 to 2017. They continued funding the Wuhan research because they weren't asked to pause what they had already started. 200 scientists from around the world came together and said, look, the mutations that happen in the lab are different from the mutations that would happen in nature. So there's no guarantee that any treatments that we develop for the lab created version will work on any of the ones created in nature. So the risk of something getting out is greater than the reward of this kind of research leading to a treatment for a future possible pandemic. In addition to the risk of accidents, there's also security risks of any workers who could have bad intentions at these labs. Now, we can say that the research that was done in the Wuhan lab helped them to predict this pandemic. However, they had already predicted that this was possible based on the metagenomics data alone. So I don't think it helped to predict it. There is medication known as EIDD 1931. This has been developed in collaboration between the University of North Carolina and the Emory Institute of Drug Development, and it was based on the research of the mouse-adapted coronavirus. We'll talk more about that in the next episode, but in the meantime, we can keep an eye on it as a possible reward of such research. In 2017, the moratorium was lifted and the US continued funding gain-of-function research. We continue to have accidents at laboratories. As recent as August 2019, we found that the U.S. Army for Medical Research Institute of Infectious Diseases suspended its biosafety level 3 and 4 operation while conducting repair to its waste treatment plant. It had been damaged for a year before, so we don't know how much could have been leaking out of there over a year time frame before they shut down just the 3 and 4 level operations. According to a survey of laboratory-acquired infections around the world, we see that reporting laboratory-associated infections has been largely voluntary, and there's no way to determine the real number of people involved or to know the precise risk of workers. Over 16 authors from around the world compiled this data, and what we can see here of the listed infections is just what they're voluntarily sharing with us. Again, it doesn't mean necessarily that they're bad, it just means that they're being honest in the collection of this information. This article talks about how some researchers prefer to do their research in other countries where they can get around red tape or any bioethical opposition that they face in the West. The study shows that they were accredited by an animal care organization. I just want to emphasize that we might want to have some international regulations on bioethics and biolab safety so that researchers don't just move their work to another country to get around it. As biotech organizations spend a lot of money influencing our government and lobbying to reduce regulations on them while increasing their funding, it's hard to know what is produced for our health or for their profit. Here we can see how much some individual biotech companies have spent in lobbying and influencing government officials. So we're less likely to hear about anything shady they're doing from either party, as they contribute to both parties in the House and Senate while also investing in the federal branch. Then Chinese investment in US health and biotech has jumped from around 116 million each year from 2009 to 2013 to $1 billion per year from 2014 to 2016. It's possible that China recognizes the risks of having too many biosafety level 4 labs and would rather invest in research abroad. So the Government Accountability Office warns against biolab safety concerns here in 2014. It was part of the decision on the moratorium. They list different organizations monitoring control of some dangerous pathogens, but some that could cause a pandemic aren't under anyone's umbrella. And they recognize that we need some national standards and procedures for all laboratories dealing in these kinds of dangerous substances. Then, in a September 2020 report to congressional committees, we can see how much we've progressed. They address duplication, overlap, and fragmentation within the program. So there have been a bunch of arguments on the organization structure. They hadn't really gotten around to much inspecting. They continue, the FDA should give the Office of Lab Safety necessary authority for oversight and ensure their independence of any influence, including funding. They also noted that there were no permanent OLS staff dedicated to inspections of facilities, even though they needed 6 staff to be able to inspect all the biolabs under their purview once every 3 years. They highlight which of the recommendations they had made in 2016 had been implemented. Of the 5, only 1 was fully implemented, even though Health and Human Services agreed with all of the recommendations. They conclude, until the FDA fully addresses the challenges we identified, the integrity of FDA's laboratory safety program and the agency's ability to ensure the safety of the laboratory's personnel and prevent the accidental release of hazardous biological agents, such as the virus that causes COVID-19, will continue to be a risk. So that's what's going on nationally. Knowing this has made me believe that we're actually incredibly lucky that this is the first time we're facing a pandemic like this, considering how many times equally or more dangerous pathogens have gotten out of laboratories doing similar research. Internationally, we can see that the World Health Organization has consultative meetings on high or maximum containment level laboratories. All of these international organizations have open discussions where they talk about best practices and challenges that they face and how to address them. There's no actual regulatory body, just advisory. In looking at what we can't know about the origins, the nature of the virus makes it hard to identify a true patient zero due to the attempts to protect nations' and politicians' reputations. And because of the biotech bribes, we can't really know the extent of responsibility. Finally, we can't know the current extent of biolab risks due to all the gaps in oversight and lack of international standards of monitoring and reporting. Now, I don't like gambling, but I love playing poker. Texas Hold'em poker is my favorite because it also shows how mutations work. In this game, each player gets 2 private cards, and the table has 5 shared community cards. Of the 7 cards you get, you choose the 5 that you think will most likely win. Now, if you get 2 aces, those are usually like the highest, best 2 cards you could get, but they're not necessarily winners depending on what randomly comes up in the community cards. So if 2 aces are like the best genes, then we look here and my genes are just a measly 4 and 10. But if we look at the community cards, we see my measly little 4 helps me get a straight and gives me a better win than if I would have had 2 aces. This is kind of how viral mutations work. They're capable of shuffling and shuffling and shuffling over and over again until what they get happens to fit in the environment better. I think trying to outmutate a virus is kind of like trying to cheat at a casino, and we know that the casino always wins. Because while we might be playing one hand, this virus is mutating in so many places at the same time. Every month we work on managing it, it mutates about 3 times to better survive among us. Now that we're facing a global pandemic, I think it's time for citizens to unite internationally and demand our governments stop using our tax dollars for gain-of-function research until they could prove that the reward is greater than the risk. I also think we need an international, independent, transparent biolab safety organization. I understand that government officials want to avoid panic, but I think we can all treat each other like adults, and maybe we international citizens can agree that we promise not to overreact if you're more honest with us, but we expect you to be honest with us about all the risks we face. Usually in science fiction stories, making chimeric hybrid creatures never turns out well. In reality, I don't see how playing with mutating viruses, which are designed to easily keep mutating to find better fits, could yield more benefits than risks, because I don't think we have the means of calculating all the risks yet. One day, maybe, if we continue collecting and inputting metagenomic data, we might get there, but I don't think we're there yet. Finally, small farms have operated responsibly and respectfully for centuries. However, factory farming increases chances of chimeric and zoonotic disease causing microbes. So please remember, every time you see cheap meat, it was a life. And if it's too cheap, it's probably bad for you, animals, workers, the environment, and global health. And the more we invade and expand into untouched lands, the more we increase human interaction with exotic animals, which can lead to more novel zoonotic diseases. I hope we can all agree that we don't want to face another pandemic like this. And gain-of-function research may not be worth the risk. Finally, the ability for the biotech industry to be able to bribe our government is bad for all of our health. While placing blame isn't productive, pointing out the problems that could have led to this mess could help us tell our government officials the kind of changes we expect them to make to keep us all safe. Thank you.